Welcome friends, welcome back again, and today I am here with a new video lecture, that is Chargaffs rule, and what this rule says about the double-stranded DNA structure. So we'll learn all this in this rule. So this rule was first proposed by Erwin Chargaff in the year 1905, and he was actually an Austro-Hungarian biochemist and was born on 11th August 1905, and later on he immigrated to the US, and there he worked as a professor in biochemistry department. And this, we know that there are double-stranded DNA and also single-stranded DNA present, but this rule was only for double-stranded DNA structure. And this rule later on helped the scientists to discover double helix structure of DNA. That was this double helix structure was later on discovered by, we know that Watson and Crick suggested the model of double-stranded structure of DNA, right? Watson and Crick. So this was later on helped them through this rule to lead the discovery of double helix structure of DNA. So as I already mentioned, that it was published in the year 1950. So what the rule says about the double-stranded DNA structure? As we know that DNA has two helical structure, right? They have helix-like structure, and there are two different strands. Now one goes from 5' to 3', and the other goes from 3' to 5', right? 5' to 3', and both are anti-parallel in direction. And within these strands there are present different nitrogenous bases. They are actually purines and pyrimidines. We know that adenine, thymine, guanine and cytosine are all nitrogenous bases, and there is some pairing between them. That is adenine always pairs with thymine, and guanine pairs with cytosine. And we know that guanine and cytosine has 3 bonds between them. Guanine and cytosine have 3 bonds between them. Okay. So what the rule says is that in any double-stranded DNA, the amount of adenine present is always equal to the amount of thymine, or the amount of guanine present will always be equal to the amount of cytosine present within it. By understanding the Erwin Chargaffs rule, we know that adenine is always equal to thymine. That means the amount of adenine, the percentage or the quantity of adenine that is present in one of the strand is always equal to the thymine that is present in the other strand. So this is equal to, okay, adenine is equal to thymine, and also the amount of guanine that is present is always equal to the amount of cytosine present in the DNA strand. Accordingly, he also mean to say that if adenine is equal to thymine and guanine is equal to cytosine, then adenine plus guanine is equal to thymine plus cytosine, isn't it? And we know that adenine and guanine together makes the purines, okay, and thymine plus cytosine together makes the pyrimidines. And what are these purines and pyrimidines? Purines and pyrimidines mean the purines are those, they have 2 ring-like structure here, okay, 2 carbon ring here, but the pyrimidines doesn't have 2 rings, they have only one ring, right? So uracil is present in RNA, but all the other nitrogenous bases are present in DNA: adenine, thymine, cytosine and guanine. Okay, so this rule says that if A is equal to T and G is equal to C, then A plus G is equal to T plus C. That means the amount of purine is present to the amount of pyrimidine. So following this rule, there are different questions that comes in different exams, okay, related to this rule. Some mathematical problems that asks mostly during exam, that if the concentration of adenine is 30% in a double-stranded DNA, okay, So what will be the percentage of guanine? This type of question always comes during exam. So what will be the solution? So here it is written that adenine is 30%, right? Adenine is equal to 30%, and we know that adenine, if adenine is 30%, then thymine is A is equal to T. That means thymine will also be 30%, right? So here see, if adenine is 30%, then thymine will also be 30%, right? Together, if we do plus adenine and thymine, that means 30 plus 30, that is 60%, right? So guanine and cytosine together will have how much? So we know that the total nitrogenous bases is 100%, right? 100%. And from that 100%, A and T made already 60%. So guanine and cytosine will make 60 minus 100, right? So 60 minus 100 will be 40%. And that means that together will make 40%. That means if we here in the question they have asked what will be the percentage of only guanine. So only guanine's percentage will be 40 divided by 2, because if we take together guanine and cytosine, then only we will have 40%. Then only if we consider guanine, then it will be 40 divided by 2, that means 20%. So these are some of the questions, mathematical problems I have also seen during different exams, that this type of question usually comes. And there are some other more problems, mathematical problems like this. I will come with it in my next lecture video series or in another lecture video series. So this is what the Chargaffs rule says. I hope you understood this, and do wait for my next video to come. And if you haven't subscribed to my channel, do subscribe and like, and also share with your friends. Thank you.